Okay, hello everybody. My name is Patrick BOLAN. Welcome to Zoom session 3 for the junior investigator poster session. We have 4 presenters here who are each going to give a talk. Let me share a couple of slides here describing the outline of this section here. Try that again. Here we go. There we go. Nope, there it is. Okay. So we're going to start here with, so in this session, the goals, what we're trying to do here is to empower our junior investigators to lead and bring different research labs who are involved in the IEM together to try and foster some sort of collaborations and maybe interconnection or communication between our labs. It's also give you junior investigators an opportunity. So we have some students and 1 postdoc here, a little extra chance to give some talking and collaboration, and that's always useful for your whole life, not just when you're a junior, folks. So this is always useful. So what we're going to do is I'm the faculty facilitator, and we have 4 presentations. You guys will each we'll go alphabetically and give about 5-minute presentations with a couple questions, and then we'll have a discussion. We'll have a few questions after each one, if there are some, and we'll have a facilitated discussion, just an open talk about what we can do to collaborate, and maybe what brainstorming things can come by looking across these projects to see what we can figure out. And of course, all this is going to be recorded. So with that, let me get started. And here's our 4 speakers. We're just going to go in alphabetical order. And I think I'll start by introducing our first person. This is Atisha Ghosh. Her talk is on automatic 2D segmentation of chronic subdural hematoma from a head CT scan. And with that, let me stop my share. And Atisha, why don't you set yours up and get ready to go.
Hey.
Well, also just, we said a little introduction, but tell what lab you're from and where you're at in your career. That'd be great, little introduction.
Okay. I'm sorry, just give me a sec. Okay. Hi, everyone. I'm Atisha. I am a graduate student in the Bioinformatics and Computational Biology program. Today, my presentation is on an automated 2D segmentation of chronic subdural hematoma from the head CT scan. I'm co-advised by Yuk Sham, who is a bioinformatics faculty member at the university, and Uzma Samadani, who's a neurosurgeon at the Veterans Affairs Minneapolis. So let's start with the clinical significance of a subdural hematoma. A subdural hematoma is the accumulation of blood on the surface of the brain. Blood and clotted blood on the surface of the brain is often the result of traumatic brain injury. The bridging veins that connect the brain to the skull get damaged, resulting in the collection of the blood. The hematoma can become chronic within a couple of days. 32% of patients die within the first year of diagnosis, and survivors are vulnerable to iron toxicity on the surface of the brain, which is associated with diseases such as dementia. By the year 2030, 60,000 patients, 60,000 cases of subdural hematoma are projected to occur annually in the United States. Non-contrast CAT scan is used to diagnose the disease. The hematoma appears generally elliptical or crescent-shaped on the scan. The current treatment is twist drill craniotomy. A small incision is made through the skull to drain the hematoma to release the intracranial pressure that gets built up, as well as toxic products. The current procedure to make the incision is to find the thickest area of hematoma from the scan and use surface landmarks to make the incision on the skull. This method is inefficient as the hematoma does not get drained completely and can result in recurrence. The goal of the project is to find the optimized drill placement location to improve patient outcomes. So the hematoma can appear bright white on a CT scan when it is initially diagnosed. This state is known as hyperdense. This is when the hematoma is more dense than the surrounding brain contents. As the blood clots, it can appear darker, and this is known as the hypodense. And the clot can break down due to enzymatic digest of the blood protein, and this stage is known as isodense. This is when the hematoma has the same density as the rest of the brain contents. The segmentation pipeline that is created has only been created so far for the hypodense hematoma. The segmentation approach has 6 steps. The first step is statistical region merging that assigns similar intensities, that assigns the same value to similar intensities that allows for easier clustering in the following k-means clustering stage. Intensity thresholding is used to pick the cluster with the k-means centroid that is corresponding to the intensity of the hyperdense hematoma. This is followed by erosion and dilation to get rid of some of the noise. This is followed by the region growing method that segments out connected structures. Pixel thresholding is then the last step that is used to pick the hematoma cluster, and also spits out the centroid of the hematoma cluster. This is our predicted, or for now, the targeted twist drill placement location. The new approach on 35 patients has achieved over 90% accuracy, precision, and recall. The predicted drill position of the approach differs from the targeted value with around a median value of like 2 millimeter and a maximum value of around 7 millimeter. When the placement tolerance, which we've currently set as 5 millimeter between our targeted spot and the predicted spot, when it's 5 millimeter, the failure rate is 14.3%. But as we increase our placement tolerance to 8 millimeter, the failure rate goes down to about 0. So in conclusion, the performance of the workflow in hyperdense 2D hematoma classification achieves 91.43%, over 90% accuracy, precision, and sensitivity. The predicted drill placement is on average 2.75 millimeter away from the targeted site. And lastly, at 5 millimeter tolerance, the failure rate is 14.3% and goes down to 0% at 8 millimeter tolerance. I would like to end this presentation by thanking our collaborators at the Veterans Affairs Minneapolis Center Care and the funding agency for this research, the Minnesota State Spinal Cord and Traumatic Brain Injury Grant Program. Thank you.
Great, thank you for that, Tisha. Let me ask you a quick question. Well, anybody else have any questions first while I'm quickly scrambling for a question?
I'm kind of curious, how much does it change size throughout the course of a year? You know, like you were talking about initially it'll appear one way and then it can become hypo-intense. Does it change in position and size as well?
Yeah, within 14 days, you can change the state from like hyperdense to hypodense. And yeah, it changes pretty fast. The growth is pretty fast.
The question I was thinking about was the stage that you were of the hematoma. So you were, this algorithm you have is targeted for that stage where it's hypodense, and that looks like the easiest stage. Is that the most important? You know, it's obviously a harder problem if you're going to make something that's going to work at the various stages. Which is, do you know the clinical need? Where is it most important that you're able to do the drill placement?
It's mostly diagnosed in the chronic subdural hematoma because most people don't really go in for like a diagnosis right after they fall. So you generally, when it appears on a scan, it's either in the hyperdense or the hypodense phase. And it's either in the hypodense or the isodense phase. And so right now we're waiting on IRB approval. So this was the most number of patients we had was for the hypodense, and that's kind of where we started. And we figured if we started for one, then it'll be easier to build off for the other ones. And this was the easiest one given hypodense appears darker. So the intensity value is already separate enough that it's a good, easy place to start, yeah.
Do you think when you actually, will you have like separate algorithms for the different phases, or have the algorithm figure out which stage it's in? Or do you imagine that you could do it with a single approach across all three different phases?
I think the stage that I would change it at would be the intensity thresholding stage.
Okay. Okay, well, let's keep moving along. Thank you very much for that. If you don't mind unsharing your screen, we'll switch to our next speaker.
Is it possible for you to unshare me?
I don't know. It doesn't.
My mouse seems to be really giving me trouble today.
Okay, well, I'm sharing mine. Maybe I can make it so. Give me a sec to figure that out.
Are you using a Windows computer? I think you can hit Alt and S and it will unshare.
Alt and S. Oh, perfect. Thank you.
Thank you, Matt. That's a great lead in. Oh, actually, you know, I see we have some chat questions. Let me take a quick look. Oh, just Dr. Odie dropping in to say hello, and we're recording, checking in on us. Great. Oh, hi, Dave. Okay, we're on to our second speaker. This is I'll share mine and give you an introduction. This is Matt Rhines. Actually, Matt, why don't you just go ahead and take over yourself there, because you'll have a better title slide than I will anyway. So.
Sure. I'm gonna start my share.
Oh, sorry, I gotta undo that. My bad.
Oh, okay, no problem. Let's go.
And yes, Dave, we're recording.
Everyone see it? Okay, so before I begin, I'm just gonna introduce myself. My name is Matt. I work in Professor Kodo Naramaya's lab in mechanical engineering. I'm a graduate student in the biomedical engineering department. And we work on technologies for neuroscience. And that is what I'll be talking about today. I'm a third-year PhD student, so. Okay, so just to begin, here's what I'll be talking about: technologies we've developed to enable mesoscale calcium, functional calcium imaging, and freely behaving mice using a head-mounted microscope. And before I begin, I would just like to give a motivation or a background as to why we are developing this technology. So most of what neuroscientists know today has been from investigations into activity from regional circuits or localized regions of the brain. But there's an increasing understanding by neuroscientists that an understanding of how all of these regions are talking to each other or coordinating to produce one behavior, and how such behavior, how that coordination changes is needed. That understanding is definitely lacking. Fortunately, there have been a lot of tools that have been developed, such as, let me get my laser pointer here. How do I do that? Can you see my mouse actually? The recent tools such as genetically encoded calcium indicators, shown on the right here, was a cartoon of one shown on the right here, have enabled neuroscientists to monitor large regions of the brain simultaneously at a high resolution. And these tools have enabled neuroscientists, they've empowered them to do experiments where we have discovered changes in mesoscale activity across large regions of the cortex, such as shown in the bottom here, which is demonstrated in a mouse performing a motor learning task, where it's head fixed here and it's trained to press a lever to receive some water. And we've discovered that these changes in activity across the cortex as the mice learn to perform the task. But in all such investigations into mesoscale cortical activity, these are always performed in restrained animals, which significantly limits the behaviors that you can investigate, changes in mesoscale calcium dynamics. And this is what we sought to this is a challenge we sought to overcome with the mesoscope, which is the technology that I'm presenting today. The way it works is we have previously developed methods to gain and maintain optical access to large regions of the cortex in mice by using a 3D printed polymer skull that interfaces optically with the brain using polyethylene terephthalate, which is a biocompatible polymer. It's actually plastic water bottle material. And we have modified our implant to include a way to fix a device using magnets to the implant and a posterior tail screw, which isn't shown in this CAD render here. The main body of the mesoscope consists of blue LEDs filtered to the excitation band of our genetically encoded calcium indicator, which is, we're using GCamp. And there are 3 such blue LEDs to provide an even illumination across the cortex. There's also a green LED in the posterior, which can allow us to measure reflectance signals, which we can use for hemodynamic corrections, which are changes in to give a short description, it's changes in optical properties of tissue with neural activity that change blood flow. Next, all of the signals are captured through a collector lens and filtered to the the emission band of our GCamp, and those signals are projected onto a CMOS sensor, which we can focus using by moving the top portion of the device and fixing it in place using a coarse focus set screw. The entire device, when assembled, weighs 3.78 grams, which I'll be showing later in this presentation is low enough to enable free behavior in mice. And photos of the device from 2 angles are shown on the right. Okay, so then after we constructed the device, we moved on to test the optical properties of it. We tested the resolution of it using a US Air Force resolution test target. First, I'd just like to point out that there are many fine details you can view. This is actually shown in a GCamp, a mouse expressing a Thy1 GCamp transgenic mouse expressing this genetically encoded calcium indicator in its cortex. And then we moved a resolution test target around 8 fields of view that represent what we see when we implant it on a mouse, and found that the resolution ranges from around 39 to 56 micrometers, which is very sufficient for mesoscale imaging. We also tested our ability to measure calcium signals. We compared it to macroscope, which the data I'm not showing here, but you can find it in the preprint that is available on BioRxiv. I found that it was comparable to conventional imaging methods, which are usually a macroscope. And we also demonstrated that we can measure sensory stimulus evoked responses across the cortex, shown by this data here. That circle is just an analysis of the peak that we used to average 20 trials, and we found a peak that corresponded to the application of a LED stimulus to the left eye. So next, we also wanted to test how much it would affect natural behavior, and we used an open field arena test to do so. Shown on the left here, the mouse is able to perform plenty of natural behaviors, such as rearing, which you just saw. We also traced the behavior in the arena and found that there was no significant difference in measures related to the agility of mice when mounted with the mesoscope. Also note that the mouse is moving around quite a lot, which can affect the imaging plane. So we are able to reconstruct the imaging plane is stable enough to reconstruct the calcium imaging using motion correction algorithms, as shown in the video on the right here. Given this, we are able to perform some of what we believe are some of the first ever mesoscale maps of activity during social behavior in mice. Just to really quickly overview the data that I'm presenting here. These mice were placed, a mouse fitted with a mesoscope is placed with a companion mouse in an arena, and then we traced its behavior using deep lab cup machine learning algorithms, and then took the correlation between multiple brain regions with the entire cortex and constructed activity maps that give us some pretty interesting information demonstrating that there is an increase in functional connectivity, increase in correlation in neural signals during epochs when the mice were interacting, demonstrated by these thick lines and these traces, and shown in the data on the bottom here, which entails an increase in functional connectivity with social interaction. And just, and that concludes my presentation, but really quickly, I would also like to thank and acknowledge my lab group and my advisor, Professor Kodan Daramaya, our funding sources in the National Institutes of Neurological Disorders and Stroke, and our collaborators in Timothy Ebner's research group, which we worked closely with them throughout every step of developing this technology. And that concludes my presentation.
Great, thank you, Matt. Wow, it's some neat tech, really cool tech with that, really nice engineering to get that thing all on there. Let me ask you a quick question so I can keep on time. When you say mesoscale, and you were about 40 micron resolution there, I look at these images, can you localize these behaviors to, the different various motor cortex or the prefrontal cortex, or what kind of resolution do you need, and do you get enough with this approach?
That's a good question. So actually, there is one bit I left out of when I was talking about this here. These experiments are usually done in mice with refractive index matching techniques applied to an intact skull, which gives a lower resolution than this. And they found that about 200 micrometers to half a millimeter is sufficient for these sort of localization of at least regional activity, and published in other papers. So with this, we're pretty confident that we're able to localize. We actually also have some other data in this preprint, and that is we hope to publish soon, where we demonstrate even more sort of a localization of at least sensory stimulus evoked responses and such, and stimuli applied while mice are undergoing free behavior.
Yeah, cool. Well, maybe we can get some more questions after, but I'd like to keep rolling, if you don't mind. Thank you very much, Matt. Would you mind unsharing your screen? It's Alt-S.
Yeah.
And let's see. And our next presentation is Sarah Saunders. Sarah, you want to share your screen there?
Yep. I will do that. Just 1 second.
Her talk is, or her presentation is multi-zonal prostate segmentation with convolutional neural networks, a comparison of 2D versus 3D UNets.
Yeah, okay, well, thank you for that introduction, Pat. I am a fourth-year PhD student in Pat BOLAN's lab at the CMRR. And as he said, we're doing work with multi-zonal prostate segmentation with convolutional neural networks. And here I'll present a comparison of 2D versus 3D UNets. So first, just a little bit of background information about this project. It's part of a bigger project with the goal of developing an algorithm to detect and diagnose prostate cancer. So prostate cancer is quite prevalent. It affects about 1.3 million people worldwide, as well as, or including, I guess, 160,000 in the US annually. But fortunately, the 5-year survival rate is quite high, at about 98% for US patients diagnosed with prostate cancer. Accurate diagnosis and staging of prostate cancer are necessary to optimize treatment. And diagnosis is typically done with methods such as prostate-specific antigen level screening, transrectal ultrasound, biopsy, which may include staging, as well as MRI. And typical treatment can include active surveillance for the least aggressive cases, or surgery and radiation if the disease is localized, or more severe cases with metastases may necessitate androgen deprivation therapy and chemotherapy. And so while these treatments can be life-saving, they do pose the risk of serious side effects, including sexual dysfunction and loss of bladder control. So because prostate cancer differs in presentation and prevalence within different regions of the prostate, I'll just go over a brief introduction to prostate anatomy here. So here in the upper right, you can see there's an axial slice of a prostate. This region in purple here on the sides is the peripheral zone. And then the entire remaining prostate is what we call the central gland for purposes of this project, and it can actually be divided into several other subregions, including the anterior fibromuscular stroma, the transition zone, the central zone, and the urethral sphincter. And for comparison, when you look at the MRI here, you can see the peripheral zone is the bright region here, and the remainder of the prostate, the central gland, is the darker region. So here's just a sample of how the different regions, the peripheral zone and the central gland, can appear at different slices of the prostate. When we acquire data, we take 19 slice scans, and you can see at the apex of the prostate, both the peripheral zone and central gland are relatively small, but as you progress upwards, towards the mid region, and then the base, they grow in size. So for this study, we've acquired MRI scans from 50 participants, all of whom had been diagnosed with prostate cancer. And as part of the algorithm to detect and diagnose prostate cancer, it's helpful to have segmentations of the prostate into these two different zones. And while it's possible to do this manually, it's very time consuming and requires some expertise. And so it's impractical. Because of this, we're developing convolutional neural networks to perform multi-zone segmentation. And for training data, I've actually manually segmented 50 of these cases using ITCaseNow. We performed image segmentation with 2D and 3D units. And first, I'll just give a little brief overview about the unit and how it can be used for image segmentation here. So this is a diagram of the original unit, which was developed by Ronenberger back in 2015. And as you can see, there's sort of an encoding arm here on the left and a decoding arm on the right. And so the encoding arm takes features from the MRI images and learns what features are valuable for predicting the prostate segmentation. And then the decoding arm looks at the MRI images and combines these features to produce an output segmentation. So when we train the model, we provide it with the MRI images, the original MRI images, as well as the manually segmented ground truth masks. And from this, it learns what features it needs to later predict segmentations on yet unseen MRI images. And you'll notice that the unit consists of several different layers here. There's the original layer here, which contains the initial resolution. And then in subsequent layers, it's downsampled. So the features that we'll learn in the original layer are finer, such as like maybe the texture of the prostate, whereas the lower levels contain coarser features. And then this work compares the 2D versus 3D units. For the 2D unit, we process information from single 2D axial slices to produce 2D segmentations. This took about 2 hours to train on a GPU. And then the 3D unit, in contrast, we utilized information from the whole 3D image, which includes the connections between axial, coronal, and sagittal slices. And then this one took about 14 hours to train on the GPU. Though it took hours to train, it can actually predict the segmentations once the model is trained within just seconds. And here's a sample of the results here that are representative of the model. So you can see a comparison of the 2D unit versus the 3D unit here. I trained it with both 5 cases as well as 40 cases, and you can see that the 3D unit far outperforms the 2D unit, particularly when few cases are used. So for example, if you compare these two here, you see the 2D unit, the segmentation of the red peripheral zone bleeds far beyond the prostate, but in the 3D unit, it's a better approximation of where the peripheral zone actually is. And so aside from visual inspection, the metric that we use to measure accuracy of the segmentation is a dice similarity coefficient. And I just have a little diagram of how that can work here on the right with sample 2D images. So for example, if you have a purple ground truth image and a yellow predicted image, the dice similarity coefficient would be calculated by taking twice the intersection of these images over the sum of their areas. And for a 3D unit, I did this with volumes rather than areas. And then in the lower right-hand corner here, you can see there's the dice coefficient for both the 2D and 3D unit for both 5 and 40 cases. And the best results were achieved with a 3D unit that was trained on 40 cases. And these results here of a dice coefficient of 0.76 for the peripheral zone, 0.85 for the central gland, and 0.89 for the whole prostate are comparable to what you would achieve with inter-rater reliability. And then I just have references there. And I will stop screen sharing. Does anyone have questions?
I just asked if I unmuted myself. Great, thank you, Sarah. It was wonderful. Let me ask, so you're focusing, this is related to the paper that we're of course working on separately. You mentioned how this is important for CAD. The reason you segment is for CAD purposes, but how can you imagine using this also for guiding biopsies? And how hard would it be to actually get something like this working in like a live guidance type of setup?
Oh, that's a really good question. I think in terms of guiding biopsies, if you have the location of the prostate directly within the patient, I could see that potentially working reasonably well. But one of the issues with prostate segmentation that I've discovered just in looking at MR scans that are acquired, maybe minutes apart, is that the prostate position within a patient will shift a little bit. you know, because like the bladder will fill or something, and then it will push the prostate down. And so that part of it might be a little bit more difficult. But I'm curious about that now, because actually when you look at biopsies, I think a lot of them, I don't know. I know like prostate cancer is more commonly diagnosed in the peripheral zone. I don't know if that's because it's more biopsied, or maybe they get a good sampling and they just find more there. But I'm curious about that. It's a good question.
Good. Any other questions from our audience? Well, we can bring up a few things when we get to the discussion section. So thank you, Sarah. Let's move on and have our last speaker, Oren Solomon. And let's see. So you have your screen up. Oren is going to be talking about deep learning-based segmentation of the globus pallidus using ultra-high 7 Tesla magnetic resonance imaging.
So,
Oren.
hello everyone. As Pat said, my name is Oren. I'm a postdoc at Norman Else Group at the CMRR, and doing image processing in that regard. And today I'm going to talk a little bit about the connection between deep learning, 7 Tesla MRI, and deep brain stimulation. So deep brain stimulation, or DBS, it's an elective surgical procedure in which a patient is implanted with conducting electrodes into the brain to specific regions inside the brain. And these electrodes are also connected to a pulse generator, as you can see in the image here. And the idea is to send pulses to these specific brain regions in order to mitigate the motor symptoms of Parkinson's disease, essential tremor, dystonia, and additional ailments such as OCD. Today, we're talking about more than 200,000 cases of DBS worldwide. So it's quite a big number, I'd say. Now, the thing is with the DBS is that successful DBS surgery is critically dependent on the precise placement of the electrodes inside the target structures within the patient brain. So here's an example of an illustration of the DBS electrode inside the target known as the STN, or subthalamic nucleus. Concurrently, the rate of DBS surgeries is on the rise worldwide. So from a surgical center point of view, this poses a procedural bottleneck. First, in robustly and correctly identifying the targets or segmenting the targets for DBS surgery, and also in the post-operative assessment of the actual position of the DBS lead with respect to these targets because of some effects which happen during surgery. Now, the anatomy between patients vary dramatically, both in the shape, size, and position of these targets. So a patient-specific planning is required, and nowadays, the one-size-fits-all solution, which is termed an atlas, is being performed, which is not quite adequate. So all of this motivates the need for a robust and reliable, fully automatic and patient-specific segmentation technique. And this is what GPNet is. So GPNet is a deep learning-based automatic segmentation of a structure known as the globus pallidus. It has two compartments, the external and internal compartments, both left and right sides of the brain. And the idea is, here is pretty simple. I won't delve into the details because of time limitations, but we train a unit or attention-gated unit in a fully supervised manner, feeding it T2, 7 Tesla T2 acquisitions alongside manual segmentation of the GPI and GPE, which were performed by domain experts. And at the end, that unit is basically able to produce a 3D segmentation of both left and right, GPE and GPI, as you can see in the lower right portion of the screen, as an example. Now, to test the clinical efficacy of GPNet, so there are additional targets for DBS, but today I'm only focusing on GP, on the globus pallidus. So from a clinical potential point of view, we've looked at a cohort of 101 subjects, 43 of which were used only for testing, 58 for training, all of which were manually segmented. Quite a tedious task. So we've looked at several clinically relevant metrics. One of them, the center of mass, which measures the overall position or position of the segmented shape with respect to the manual segmentations. And you can see here, for example, in the leftmost column, these are the errors with respect to the manual segmentations of GPNet compared to state-of-the-art atlas-based segmentations. This is what's being done nowadays in the clinical setup. And you can clearly see that not only do we achieve systematically lower errors, but the distribution is also smaller compared to the atlas-based segmentations, which indicate that we are basically more resilient to patient intra-patient variability. And we've trained and tested both on healthy subjects and people or subjects with PD and essential tremor. On the right here, we can see the DICE score. And again, we see the same thing over again. We see that GPNet systematically achieves higher DICE scores compared to the atlas-based segmentations, which again is a good thing. Higher DICE means higher overlap between the two shapes, manual segmentation and the output of, in our case, the network. And lastly here, we've also estimated the volumes obtained from the different segmentations. So first, we estimated the volumes from the manual segmentations. This is the leftmost column here. In the next column, we see the volume estimates produced by GPNet, and these are the atlas-based segmentations. And you can see two things here. One, that only GPNet is able to achieve robust, basically robust estimation of the true statistical distribution of, this is the GPA and this is the GPI volume estimates. So not only that, but even if we look only at the manual segmentations, we can clearly see there's great variability between the volume estimates between different patients, meaning that, again, a one-size-fits-all solution, which is what's being done today, is not really applicable. It's not really good. And the patient-specific technique can truly capture this variability and give us a tuned solution per each patient. Lastly, I just want to show you to demonstrate the clinical potential of using this kind of technology in the clinical scenario. And in the upper panel here, you can see a reconstruction of a bilateral DBS implantation of a PD patient. So these are the DBS electrodes implanted into the brain. And in darker gray are the DBS contacts, which are inside the GPI. So here we can see the manual segmentations of the GPE in green and GPI in yellow. And the idea is to place these contacts inside the GPI. And you can see an excellent match and very good clinical outcome in this case. In the lower panel, you can see the same electrodes with respect to the automatic segmentation performed by GPNet and the GPI in blue here. And you can see again an excellent match between the electrode location, the contact locations, and the GPI itself. So this is the power of combining 7 Tesla MRI and deep learning modern techniques in order to achieve clinical efficacy. That's it. Thank you.
Great. Thank you, Oren. Wonderful. Let me ask quick first about the metrics. You showed volumes and DICE and what was the other one? Oh, the center of mass. And you know, this is something that with Sarah's work and her other projects, we talk quite a bit about what metrics are most important. And so for the goal, everybody uses DICE as kind of the default thing, but when you're trying to put that thing in the middle, which is the most clinically relevant metric, and is there some metric that would be better that would need to be developed?
So I guess the answer basically can be separated into two parts. One is what are we training on, and the second is what is the metric on which we assess the performance. So in terms of assessing the performance, I'd say from the DBS point of view, so you have two metrics which are very important. One is the center of mass, because it's the general location of the shape. These shapes are mostly ovalic in shape, I'd say, STN or GPI, and it's very important for the clinician to know roughly where the shape is. Now, in addition, I didn't show it here, but we've also measured the mean surface distance between the segmented shape and the ground truth. And the mean surface distance gives you more of an estimate of how well do we capture the overall shape. So DICE score essentially captures all of that together, but it's not something that clinicians typically look at. Or it's not, it kind of captures everything together, but it's, I'd say, all over the place. The center of mass and the MSD, mean surface distance, give you these two distinct criteria which we can translate directly to the clinical setting, because we need to know where exactly the borders of, let's say, the GPE and the GPA ends and the GPI starts. This is very important clinically. And we can also validate that using microelectrode recordings. And the other thing is the center of mass, which is the general place, the general location within the brain. Now for training, I mean, I guess as a philosophy, you'd want to train on what you assess your network, right? So This is what we kind of did. So I didn't talk about it, but the loss function is actually a composition of two losses. One is the Tversky loss, which is just an extension of the dice coefficient. It's essentially it's dice extended where you can give more emphasis to the false negatives or the false positives. And there's something which is known as the Hausdorff distance, which is it closely relates to the mean surface distance. Basically, it's a more strict criteria than the mean surface distance. Most people don't show it in papers. A few do, but most don't. But we use it. I mean, there are a few papers nowadays which actually incorporate Hausdorff distance, or MSD is a surrogate for Hausdorff distance in the optimization of the network.
Okay, thanks. Yeah, metrics are always fun when it comes to these things. Lots to think about. I'll tell you what, so where are we at? We're at 1:40 right now, so we have some time to discuss potential collaborations, but I'd kind of like to open up a little bit because we go fast through that stuff and I took all the questions. So do you guys have questions for each other? Maybe it's a good time we can start with some of those.
I had one question for Sarah regarding the, so when I saw in the slides, so the training was performed on 50 patients, but all of them were positively tested for prostate cancer, right?
Yeah.
So the question is, have you considered also feeding the network, training the network with people which were negative? Because I mean, I guess at the clinical setting, you get a scan, you don't necessarily know if the patient has or hasn't. And this might bias the network if you're training only on one type of I guess, prognosis.
Yeah, that's a really good question. I actually have considered that. It happens that the data we have was acquired from people who do have prostate cancer. So I just don't have a data set.
Oh, I see.
How many people would go in and get an MRI of the prostate willingly if there's no indication for it. That's just the bias in sampling of the data that we have.
Okay. I understand. Yeah, it's hard to get good data. I mean, that's the part, I think.
Other questions? I guess I'll tell you what. Let's just. I have a couple of things I'd like to start with, and then if you guys go back to, if some technical questions come up that you want to ask, just jump in with those things. For starters, Matt, in your work. You're near the end of your PhD, it seems, but I mean, I'm familiar with it at the center, at our lab, the Center for MR Research. There's a new optical group there, and I see like Jan Zimmerman and Prakash. I don't know if you know these guys, but they do. Jan is a relatively new faculty member, and I see he doesn't have anything here at IEM, which is too bad, because they're putting these kinds of larger scale devices for doing two photon optical stuff on macaques and behaving macaques. And I'm curious, first off, are you aware, have you seen those groups and seen the kind of stuff they're doing? Because there's a lot that parallels, is my first thought.
I actually have not, but that is a lot similar to what we were doing, especially with the actual implant that the mesoscope is based on was originally designed for 2-photon imaging in a head fixed animal, but probably the same case for a macaque, right? Yeah.
Well, I mean, we're an MRI center. We used to be. Now we do a lot of optical, but they wanted to get an MR compatible one too. Obviously, you can't have they can't behave freely when you're in the middle of a bore of a scanner, but you do have behaving monkey studies that, which always sounds funny when I say that out loud, but they can be trained just like the lever thing for the rat. They do tons of stuff with that with the macaques. It's quite interesting. But related to that, I remember, so Jan was showing me some stuff where he was doing a lot of posture analysis. So he has his animals roaming free in a cage, and they're trying to look at behaviors and stuff. And deep learning, I see a lot of people doing posture work. Have you done that with your mice, doing their rearing and their interaction with each other? Is that how you got those traces?
Yes, that is the next step of our research, where we're trying to develop more sort of computational behavior analyses to go with because we're going to generate a lot of data when we get this mesoscale dynamics. And there's sort of two things about this. Anyone doing it right now, we would be open to working with them in any way possible. And then as a second, we have some projects that I actually, I was going a little over the time, so I didn't mention, but we have some projects ongoing. For example, in Timothy Ebner's group, they're using the mesoscope in a behavioral paradigm for novel object recognition in a freely behaving mouse. And actually, my PhD work is in a behavioral paradigm where we are looking at the evolution of mesoscale cortical activity for mice learning to navigate a space. And that's going to require a lot of, for the mouse is going to be, there's going to be a lot of information related, not related to the mouse actually just navigating and learning, but also just posture for one thing, and even stereotyped movements, and any computational behavioral analysis tools are really powerful in that regard. They're better than manual scoring, which is what a lot of animal behavior people have been doing for years. So just throwing all that out there, yeah.
Yeah, I was, as I think about, maybe I should introduce you guys, but Tim Ebner he works with Jan on stuff too. So if you work with Dr. Ebner, maybe it'd be a good group to reach out to say, hey, there's other folks on campus to connect with in that way. So he'd be a good resource.
Right. Yeah. Thank you. Yeah.
Yeah.
I agree.
Matt, I have a question for you, a far less technical question than some of the others maybe. So when you put these devices on the mouse's head,
yeah,
how do you get it so they don't just try to whack it off?
That required a lot of design changes, because they actually some of them would learn that they can sort of like pry it off if we just had the two magnets on the side or something. And also getting it on and off, they begin to sort of associate, there's a protective cap part of it that isn't shown in this presentation, but when that comes off, they begin to immediately, they associate it with me putting a heavy weight on their head, which they don't like at all. So they're trying to get it off. So that's why we have this screw portion. I didn't show it in my presentation well enough. There's a screw on the back of the implant that we actually fix it on there. So even if they try, they probably learn that they can't get it off. So they just begin to try to do everything as usual.
Okay, so they just kind of let it be then at that point.
So I do have more questions, but I think I should not keep asking questions because I could do that too much. I mean, part of the thing we're supposed to be doing here is maybe sort of talk about how we can imagine some collaborations. I mean, obviously suggesting that between Matt's team and Jan's stuff, but other groups. I mean, obviously the stuff, Oren, you're doing, and sorry, you guys are both doing 3D UNets, and I saw, Oren, yours has 3D attention stuff, which I don't really know what it is. I've seen that in the literature, but I'm curious, maybe you guys could have some comments about which of you see something in your project that you think could help one of the other people, or vice versa. Like, hey, I could, like I'm thinking, hey, sorry, we should find out what I'm pointing here because I think Oren is right here on my screen. That we should find out what his attention unit is, and maybe that's useful for us. But I wonder what you guys think about that. Maybe I could start with Atisha. We've talked more than you, so I wonder if you have some comments or thoughts about how your project would cross over with some of the things we have here.
I think when you said the neuro navigation thing, I think the project between me, Oren, and Sarah, all three of us have a neuro navigation component in terms of like the drill placement for all three of our projects. So I feel like finding out some way of creating like the scatter points to like a CAD model, something along those lines might be of interest to all three of us. Yeah.
Have you? So I saw you're using those image processing methods. Have you looked at the UNet type structures, or have you done even with the conventional method that you're doing? You're doing 2D with the slices back and forth. Your approach. Have you tried to extend to 3D?
I have tried to extend to 3D, and so this approach was kind of this will now be optimized for the 2D version, and I've been going back and forth between 2D and 3D, but I'm yet to scale this particular approach to the 3D. So my next goal for now is to scale it to the 3D and also use convolutional neural networks to see if the segmentation improves. I'm taking the machine learning class right now, and one of my project goals for the class is to use AlexNet, because I'm new to this field, so that's what the professor recommended using and kind of playing around with the data to see the segmentation. But currently, what I have scaled to 3D and the convolutional neural network, comparing it, like if it's any better. Yeah.
Related to that, what's the processing time? Because you are probably in a situation where you want to get some results fast, right?
Mm-hmm.
How long does your algorithm currently take?
For all the slices, it actually does not take more than like maybe 5 minutes, like 35 patients, single slice per patient. I ran it for all the files at once, and it takes about 5 minutes to give the centroids, yeah. So it does not, in 2D, it does not take that long. I'm assuming in 3D, it would be a little bit longer. Yeah. I do have a really good graphics card, so that probably helps.
If you were to do it in 3D, is there a way you could do like a rough estimate in 2D and then narrow down the scope of your like field of view so that it's faster in 3D?
We were thinking about it. We're thinking of using the biggest slice because the hematoma would be the biggest in some of the slices. So we were thinking of using the biggest slice and then maybe using 2 or 3 slices from the biggest slice and reducing the number of slices we're processing. But right now the challenge would be how do we automatically identify the biggest slice? Because right now I'm manually picking the biggest slice. Or like if the radiology report has the biggest slice or the slice numbers, it'll be easier to just mine those. But I have not seen like actual patient records yet, so I have no idea what the records have, like what kind of information they have, things like that. Yeah. Do you have any suggestions?
Nothing in particular. I was just wondering, like, if, you know, even if you do your 2D and if that runs really quickly, and then say your 3D takes significantly longer, but you need fast results, if you could do your 2D model and then use that to maybe find like the biggest slice or just a region that you can focus on, and then say, like, oh, I'm going to do this region, and then use that and input that to the 3D so it has fewer voxels or pixels or whatever to search for.
That makes sense. Okay, yeah, I'll try that.
And I don't know if it'll work or not. It was just a thought.
I mean, it's worth trying and seeing. I mean, that's the best part of research, I think.
I had a general question kind of for the group. So first one who unmutes themselves gets to answer or say something about it. I mean, one common theme I saw that where everybody's doing, I mean, different things, some guidance, but one thing that most of us haven't just said out loud is motion. Sarah, you mentioned the prostate moves. It does, I know that does quite a bit. Atisha, I'm sure from between the CT scans go so fast, but between when the CT, I don't know if people are in a stereotactic frame or if motion's an issue. I know, Oren, with your stuff, the brain squishes when you put that stuff in there, and position is very difficult. And Matt, with those mice moving around, I'm sure, I don't know how rigid you are there, but so of all these, are they all image processing targeting types of problems? How important is motion? And is, you know, for the first order, everybody starting in these areas and all these projects, assumption number one, well, let's assume everything stays still, right? And then you do all the thing and get it working. And then how many of you are having problems with motion and what solutions are you coming to? That's my question.
Yes, my mic is on, so I can start. So motion. So right now, motion is not a factor in my case because, as far as I know, I don't do the acquisitions on the machine. So we have other guys in the group who do them. But first, the patients which are PD patients and essentially people with tremors, they are on medication. So it mitigates to some degree the effect. But what we do in the group is it's an optimized acquisition for these deep brain structures. So we don't acquire the entire brain. We only acquire a slab. It's a 2D acquisition of a slab of the brain. So it's relatively fast. I mean, sometimes the acquisition might get fuzzy. And so we discard this acquisition and do it again. But for most of the time, per modality, I didn't see any motion artifacts. We do need to register between like T1, T2 SW acquisitions. We do have to register between them. But per acquisition, right now I've been working only with T2 acquisitions, and so we don't have that issue. As to what you said about when we open the dura of the skull, yeah, there's this what's known as the brain shift, everything squishes. This project doesn't deal with that at all because it's only for the pre-planning stage. So we don't have any kind of these problems. And 4 weeks later, when you want to assess the post-operative placement of the lid with respect to these targets, after roughly 4 weeks, the brain pops back to the right position, usually, I hope. And then you can do you can just place them together and see. And this is like the last figure I showed you. But yeah, the intra-op scene is something we definitely want to look into it, but it's, as far as I know, it's mostly uncharted territory in DBS.
If I may go next about this, the motion for my project is such a huge deal. And some of the things Oren was talking about, another thing I left out of this presentation, that it's in the bioRxiv preprint. We actually are doing a large craniotomy to put this implant on. And we observe, and even in mice, the brain will move right after you do the craniotomy. And depending on the quality of the implant, or sometimes just the individual mouse, you can get a brain that will sort of swish around in its non-rigid movement as well. Some regions will move more than others in all 3 dimensions. So in cases where we cannot reconstruct a stable signal, which even that alone, we're not even entirely sure if we're only sure to the extent that these algorithms have been previously used. If we can't reconstruct a signal, we actually can't use the data from that mouse. But the positive from that is that there are other things, there's other data we can get, which isn't the project I'm working on. Since we are imaging the cortex, there are other things that people are interested in, changes in vessel diameters and such. Or in the case of Atisha's project, if something is ever moving on to an animal model that you are doing, we're actually using an automated surgery platform, which uses sort of fiducial components on the animal's skull. And you can image things like, I know my advisor I forgot whom exactly, was looking into visualizing the development of tumors, but any sort of like hematoma, you could probably image that as well, given you do a clean surgery. A lot of things I was thinking about just in the back of my head, but that sort of relate to the stability of your imaging field. But yeah, just to answer that question, it is a big deal with that paper. We actually just can't use data we can't reconstruct. So.
Any other motion-related comments?
I have just discarded it for now, any patients that had artifacts, but I'm hoping, like, once I have more patient data, if I see it being a problem, then thinking about it. I probably should start thinking about it.
Well, when they go to put that hole in there, I mean, you had these accuracies. If you said it was within, I think it was 8 millimeters, you had 100% accuracy. Mm-hmm. When you have a hematoma that's that big, does it kind of matter? How clinically relevant is it that you're really spot on, or do you just need to get close to the center?
So one of my advisors is a neurosurgeon, and she was fine with the 5 millimeter threshold. So I would say 8 might be pushing it, but I think 5 would be okay. But I need to see if there's like literature reviews. or if there are numbers. But I feel like if a neurosurgeon who does it on a daily basis says 5 millimeters is okay, I would think it's probably okay.
How do they pick the spot now? Yeah, see, I'm taking up my own questions instead of doing this collaborative thing. Hey, you know, I tell you, let's see, it's 2 o'clock now, actually, and I want to be respectful of everybody's time. I'm really grateful for everybody presenting and talking today. Let me share my screen real quick, and I just want to bring up one last thing. So there is this pilot project competition, and the IEM, I mean, they get a lot of money to help trick people into working together, and it really does work out. Lots of good stuff comes of it. So there is this relatively, you know, it's a modest little competition, a little pilot project, a 2-page proposal. It's due in just October 12th, just a couple weeks off from now. And there's $5,000 that can be used for something. I was commenting earlier, that's a couple Titan cards, but there's a link to it. I put the link in the chat. I sent you these posts, this PowerPoint slide the other day too. So I just want to make you aware of it. You guys all have contacts for each other, and I want to encourage you to think about stuff. And if you have questions, or if you want to follow up with each other on questions, you can include me on it if you wish. You don't have to, of course. You guys all have each other's contact information, and it would be really great if something were able to come out of that. So keep it in mind, share that information with your lab mates. And the whole point, you know, especially in these virtual times, I think one of the goals of this kind of stuff is to just connect better and say, hey, that group over there is doing something neat. You should talk to them about this sort of idea. So the more we can do that, the better. Don't be shy. We have to be less shy than we normally would be during these times, I think. So with that, I suppose I'm going to stop recording and close. And thank you guys all again for presenting. It was really great. And have a great day. Thanks.
Thank you. Bye.
Thanks, everyone.